Now guys, we will discuss unit 2, membrane physiology, nerve and muscle. The first chapter of this unit is the transport of substances through the cell membrane. Now, in this paragraph, they have discussed the extracellular and intracellular fluid composition and characteristic, which we have discussed in chapter number 1. So they are simply saying that the extracellular fluid contain sodium, calcium and bicarbonate in high concentration, and the intracellular fluid contain potassium, phosphate, magnesium, proteins and fats in high concentration. So the difference between extracellular and intracellular fluid, this is we have discussed already this in chapter number 1. So take a look over this is easy. The cell membrane is a lipid bilayer with cell membrane transport protein. We also have discussed in chapter number 2 the structure of the cell membrane, that the cell membrane consists of lipid bilayer with proteins and carbohydrate, and the protein which are present in the integral protein, they are acting as the transport proteins. So for the diffusion can occur through three routes. It can be occur through a carrier protein, which is called facilitated diffusion, or it occur through the channel protein, which is in the case of water soluble substances, and which is water insoluble. So they will the diffusion will occur through the channels. If a substance is lipid soluble, then it can directly traverses the lipid bilayer. So this is the three routes of the diffusion: two through the proteins, through the integral protein, one is directly, and the activetransport need the carrier proteins. Activetransport cannot be performed by the channel protein or directly. So the activetransport, the only means for the transport of substances through active process, is the carrier protein. So these are the cell membrane, and these are the transport carrier proteins. Now diffusion versus active transport. What is diffusion actually? We are familiar with the definition of diffusion, that the substances which goes from a high concentration to a low concentration, this is called diffusion. But what is the mechanism actually? What is the mechanism of diffusion? For example, they have discussed this in this paragraph. If this is one molecule, then this molecule, if they moves randomly from one place to another place and strike one another, so this is the moment of molecule. They have shown that it is randomly striking one another, and through this striking force they diffuse from one area to another. So the random movement of molecules which occurs in the liquid and gases, through these random movements of molecules in striking of an area, through this striking they produce a pressure, and this pressure is then responsible for the diffusion of substance from one place to another place. So this was the diffusion. Activetransport, the activetransport is which occurs against a concentration gradient and which requires energy and transport proteins. But in the case of diffusion, only the transport protein is required for the facilitated diffusion, and the channel protein is necessary for the transport of water soluble and water substances. So the activetransport, in simple words, the diffusion needs no energy for its transport because it's in high concentration. So if it is present in high concentration, through these random moments and striking, they will create a pressure and will diffuse from high concentration to low concentration. But in case of activetransport, the substance is in less concentration. In the case of activetransport, the substance is in lower concentration, so the pressure will not be so enough to move from one place to another. So in this case, they will need a carrier protein and energy. So through the expenditure of energy, this activetransport will carry molecules from low concentration to a high concentration. So this was the diffusion versus activetransport. Now diffusion, they simply discuss this phenomena of diffusion. This paragraph, that how the diffusion occurs through the molecular mechanism. And another thing, one point, another is that the scientists call this is the movement of molecule. The scientists call them what? Heat. So whenever the molecular motion, the molecular velocity is high, then the temperature or heat will be more. So this is simply they have discussed in one paragraph this entire diagram. Now diffusion through the cell membrane. Diffusion which occurs through the cell membrane depends on which substances, depend on which factors. First of all, the diffusion which occurs depends mainly on the three factors. The first factor is the amount of concentration, how much the substance is present, the amount. Second is the velocity is low or high. And the third is through the places through the pores and the places through which these particle will diffuse from this side to this side. So how many pores are present in this area? For example, if this is a barrier, and if the substance has to move from this place to this place, so it depends, the diffusion depends how much pores are present in this barrier and what is the size of these pores. So three factor. First factor is the substance, in how much the substance is present, how much is present on this side, how much is the velocity of the substance on this side, and how much pores are present in this barrier, and what is the size of pores. These three factors are responsible for the diffusion from high concentration to low concentration. If the substance is more, if the velocity is high, and if the size is large and the pores are in large concentration, so these factors will contribute to diffusion, and the diffusion will high. So these things are directly proportional to the diffusion. They have discussed this. Now this was about the simple diffusion, that the simple diffusion depends on three factors which I have discussed. But in case of the facilitated diffusion, it needs carrier protein. For example, the carrier protein will attach some substances to this place, and it will move the substance from high concentration to low concentration in this manner. So diffusion of lipid soluble substances through a lipid bilayer. The substances which are lipid soluble, they will pass through the lipid bilayer directly, and their diffusion is very rapid. So for example, the oxygen which is lipid soluble, the carbon dioxide which is lipid soluble, the alcohol which is lipid soluble. These lipid soluble substances depends on their solubility in the lipid bilayer. So the substance which is more lipid soluble will diffuse more rapidly, and vice versa. Diffusion of water insoluble substances cannot occur through the lipid bilayer because it is water insoluble. Diffusion of water in other lipid insoluble substances through the protein channels. The cell membrane contain the integral protein. For example, if this is the cell membrane, and this is the integral protein, so a channel or an opening will be present through the entire length of this protein. This is called what? This is called a pore. For example, we have aquaporins, which are the pores particularly for the water. So these pores will carry those substances which are water soluble, and these pores also depend on the size of a substance. If a molecule is large, and if it has to pass through these pores, then it will pass the diffusion rate will be very slow. For example, they have discussed one example that through the red blood cell, the water which passes through the red blood cell is 100 times more as compared to the volume of the RBC per second. So such a high amount of water is passing through the red blood cell on either side. The water diffuses through this red blood cell, and then it is coming out of the red blood cell. So the net rate of incoming and outcoming is equal. Another example that if something is a little bit in large size as compared to the water, so its diffusion rate will be very slow. For example, the urea is only 20%. The urea size is only 20% of the water, but it is 1000 times less permeable as compared to the water. So the lipid soluble substances depends on the solubility of the substance in the lipid, and the water soluble and lipid insoluble substances depend on the size of the molecule which has to pass through the pores. So diffusion of through protein pores and channel, selective permeability and gating of channels. We will discuss the difference, what is the protein pores and what is the protein channel. We will discuss two main difference. First is pores. protein pores, protein channels. The first is the protein. The the protein pores are the these are the tubes along inside the integral protein. Tubes inside the integral proteins. Inside integral protein. And they are always open. The protein pores are always open. Depends on the size and electrical nature of substances and electrical nature of substances. First of all, these are the tube inside the integral protein. Second, they are always open. And third, depends on size and electrical nature of substances. Now, the protein channels. The protein channels are actually these are the spaces through the cell membrane which are surrounded by the protein complexes. So, spaces through the cell membrane, spaces through cell membrane surrounded by complex of protein. Surrounded by complex of proteins. Second, tube inside the integral protein space through the cell membrane surrounded by complex of proteins. Always open. The protein channels are not always open. They depend on the conditions and environment of electrical charges or chemicals, and they are closed or open by gates. Not always open. Not always open. And open and closed by gates. Open and closed by gates. The third point is it is highly selective. Protein channel, it is highly selective and specific. Specific for a particular substance. For a particular substance. For example, potassium channels. These are highly specific for potassium, and sodium channel, which are highly specific for sodium channel for sodium ions. So these are the three main difference between them. If someone is confused in the last point, for example, depends on the size and electrical nature of substances. If you have noticed, the substances, these pores are highly, these are also specific. But if you have noted that the substances which are water soluble can also pass, which are water soluble and which have electrical nature which are matching to the water, can pass through these pores. But if you have noticed in this point, it is only highly specific for a particular substance. For example, potassium channels can only be responsible for the potassium, and the sodium channel is only responsible for sodium. Potassium channel is, for example, 1,000 times more permeable for the potassium than for sodium. For example, I will show one channel. They have showed one side of this channel. So this is the tetrameric. Potassium channel is tetrameric. They contain 4 units, 2 on this side and 2 on this side. So the 4 units surrounded the potassium channels, and these potassium channels, from the loops of this potassium channel, these are carbonyl oxygen. Carbonyl oxygen is attached to these places, which when the potassium ion comes to this place, then they will shed the water from the potassium. The carbonyl oxygen will shed the water from the potassium channel and will allow the potassium channel to pass through it. But in case of sodium, the sodium the sodium is very much small. For example, if this is the loop, this is the loop, and this is the sodium channel. You have noticed the sodium channel cannot interact with this carbonyl oxygen. Why? Because the sodium ions are very small and they cannot interact with both of these loop with this selective filter. This carbonyl oxygen which is present in the loop and the opening of this channel, this is called the selectivity filter. So this selectivity filter can interact easily with the potassium channels, but this selectivity filter cannot interact with the sodium channel because of the size of the sodium channel. The size of the sodium channel is not large enough to interact with both of them. So that's why this potassium channel exclude the sodium and permit the diffusion of potassium. That's why the potassium channels are highly permeable, 1,000 times more permeable for sodium is compared for potassium as compared to sodium. So this was the potassium channel. Now if we come to the sodium channel, now for example, if I draw sodium channel, if this is the sodium channel, if this is the sodium channel and this is the cell membrane, this selectivity filter was lined by the carbonyl oxygen. But in case of the sodium channels, this selectivity filter is lined by negatively charged amino acid. So they will attract the positively charged sodium ion and will repel the negative charged molecules. So this is the selectivity filter of sodium, and this is the most crucial channel. That if there are a large amount of ions and if different ions are present in this area, then this selectivity filter is highly specific for sodium, that in such a high communities of ions they can select and filter the sodium ions. So this was the potassium channel, this is the sodium channel. Now it is done. Now another important topics about these channels is the gating of protein channels. Now what is the gating of the protein channel? First of all, we have two types of gates: voltage regulated gates and ligand or chemical regulated gates. The voltage regulated gates are those gates which are regulated by the ions, by the differences of ions. For example, if we take the example of sodium channel, the sodium channel contain two types of gates. One is the activation gate and another is inactivation gate. Sodium activation gate will open only if the ionic distribution inside the cell is changed. For example, the normal resting potential of the membrane is minus 70 millivolt. If this is changed to minus 55 millivolt, only a little bit, then this activation gate will open and it will permit the sodium ion to pass through this to these channels. But if this voltage is raised to plus 35, if this is raised to plus 35 for the large neurons, then the sodium inactivation gate will closed, and it will prevent the further diffusion of sodium ions. So this was the example of voltage regulated gate or the sodium channel. And another voltage regulated gate is the potassium gate. So for the sodium has two gates. One is activation gate and another is inactivation gate. These two gates are regulated by the differences of ions. Another potassium gate it is also regulated by the differences of ions. For example, if this depolarization has been occurred, then for the resting potential, the potassium gas has to open to regain its normal resting membrane potential. So it is also regulated by the ionic distribution. Now we will discuss the ligand or chemical regulated gate. The ligand or chemical regulated gates mean that this, for example, if we take the example of acetylcholine receptor, this is for example the acetylcholine receptor. For example, if this gate, when the acetylcholine is released by the neurons, the acetylcholine will attach to this gate, and the acetylcholine through its different mechanism will change the conformation of this gate and will open the gate for the transmission of action potential. So this acetylcholine receptor this is the example of the ligand or chemical regulated gate. This gate is controlled by the chemical, for example, the neurotransmitter. And these gates, the gates of the sodium and the gates of the potassium, these were controlled by the ionic distribution. That is why this is called voltage regulated gates, and that is why this is called the chemical or ligand regulated gates.